Okay, I heard that you had some problems with understanding the regulation of transcription. In order to explain it better, I will use some video that I did not create it. I just found it on YouTube. Transcription Regulation by Flaming Manzana. You can find it on the YouTube. So I will open it, but I will add some comments from myself. That's why I'm creating the separate video, not just posting the link. Some things I will get faster and I will highlight what is the most important. So let's start.
Without it, life as we know it would cease to exist. DNA transcription is the first and most important step in a series of events required for the synthesis of essential cellular proteins. Based on changes in the environment, a cell uses different regulatory mechanisms to adjust the rate of transcription. It is the
Okay, so now, critical thing. Now we are starting about regulating of transcription. So you already know what is the transcription. Now we need to now we need to learn how it is regulated. So what is making it happen? Yes, when it happens, how fast it happens. So it is all about regulation. When, if, and how fast.
Balance between all of these mechanisms that controls transcription and ensures cellular survival. Transcription is the process of creating a single-stranded RNA from the double-stranded DNA template. The primary protein that catalyzes transcription is RNA polymerase, which is the core of the transcriptional machinery.
So one more time, this is the RNA polymerase. Yes, this is the most critical enzyme. So obviously, if something is critical, if something is the most important enzyme, it will also be very important in regulation. Because if it would not attach to the proper place, if you do not start its work, the transcription would not happen. So if we control the the RNA polymerase, we also control the transcription.
RNA polymerase is typically actively recruited to DNA during initiation of transcription. Once recruited, RNA polymerase moves along the DNA and creates complementary RNA. This step is known as elongation. Finally, when RNA polymerase...
So now we are just talking about typical transcription, yes? At this moment there was nothing about regulation. We are just getting about transcription that you already know.
...reads a signal to stop transcription or is interrupted, termination occurs. In an actual cell, transcription is not simple. The regulation of when, where, and how often transcription occurs is influenced by a number of different components. Proteins, known as transcription factors, bind to the DNA. Depending on how and where they bind, transcription factors can help initialize transcription, or they may bind to DNA in a way that blocks transcription.
Okay, so this is critical thing, yes? So we have some substances called the transcriptional factors. And these are usually proteins that can bind, for example, to DNA. Not only, but generally, when they would attach to DNA somewhere, it may increase the probability that transcription would happen, or it may decrease the probability that transcription would happen.
These functions are known generally as activation and repression.
And obviously, activation increase, repression decrease.
Transcription factors in an activating role help to orient the DNA or recruit proteins to begin transcription. These proteins have a binding affinity and structure that positively affect transcription. Transcription factors in a repressing role may block or occlude the binding of the transcriptional machinery or other transcription factors. These proteins have a combination of binding affinity and structure that negatively affect transcription. Transcription factors have DNA binding domains, which are the sections of the protein that actually interact with the DNA.
Okay, these are maybe not critical, but general DNA binding domains, when something is called binding, yes, it is a place of attachment, yes, so DNA binding domain of the protein is a part of the protein that is attaching to the DNA. Just for you to know what the author is talking about.
These binding domains are what give the transcription factor an affinity for a specific sequence of DNA.
Affinity is the propensity to do something, yes? In this case, affinity is how well this protein is suited to attach, yes?
They have a higher probability of binding and staying at a section of DNA more complementary to their binding domain. The binding of a transcription factor to its preferred sequence is not guaranteed. Many factors with different structures and affinities compete for interaction with the DNA.
Okay, and couple times also the author told the word affinity, yes? So one more time, what is the affinity? Affinity is how well something is suited to attach, in this case to DNA, for example, yes? So if something has a high affinity, for example, some protein, some transcription factor has high affinity, it means it wants very much to attach somewhere. If it has some low affinity, it is less ready to attach. So if something has high affinity, okay, let's make an example. You have two substances, yes? Transcription factor A and transcription factor B. Transcription factor A has high affinity, transcription factor B has low affinity. So now imagine that we have both of those transcription factors, A with high affinity and B with low affinity. And what would happen if there would be both of those transcriptional factors that has the affinity to the same place of the DNA, so that they want to attach to the same place in the DNA, and they are in the same place in the same time, yes? So obviously, if something wants more, with highest affinity, it means that it will attach and not the transcriptional factor with lowest affinity. Yes, so the higher the affinity, the higher the probability that it will attach in case of the competition with some other substances. Yes.
This creates a stochastic system where binding events appear random, but are, in fact, determined by the local molecular environment. Another element in the complexity of transcriptional regulation is the packaging of DNA. DNA is not flat and straight. It is packaged.
Okay, as you remember, DNA is organized. Yes, it's highly organized in highly organized structures. Those yellow balls that you can see are the histones. So, author also will go on with this topic, but just to highlight, this is very important. So, the structured DNA, how much it is coiled, how easy it is to get to the DNA is critical, because imagine that the RNA polymerase would try to get into the part of the DNA that is coiled around the histones. Yes, it would be extremely hard to manage. It's nearly impossible. So the DNA needs to be prepared. If it's too dense, too coiled, it's impossible. Yes, so when something is coiled, when the part of the DNA is extremely densely packed, this part of the DNA is blocked from the transcription. And this is one of the ways how to control it, yes? So the transcription would not happen if the DNA is too densely packed. Of course, the organism has the way to change it, yes? So if something is densely packed, it can unpack it and then start the transcription. So it's reversible. But we need to remember that the structure of the DNA is also a very important factor that regulates transcription.
In a complex known as a nucleosome. Nucleosomes allow it to pack more tightly in the nucleus. This tight packaging makes the DNA less accessible to transcription factors and transcriptional machinery. The protein that binds to DNA in a nucleosome is called a histone. Histones can be chemically modified to alter the stability of the complex and thus the strength of the interaction with the DNA.
Yeah, so obviously, the histones are the molecules that control this packaging, how dense is this structure. So there are some substances that will affect the histones, so they will affect the structure. this is, I suppose, quite easy to understand.
The combination of nucleosome position and histone binding strength influences the accessibility of a portion of the DNA to other factors. The transcriptional machinery can also affect the local environment. Once the transcriptional machinery forms a strong bond with the DNA, it can move along a strand and alter the binding of nearby transcription factors and histones. Sets of transcriptional machinery can also interact with one another. For a long segment of DNA, more than one set of machinery can be actively transcribing at the same time. Several sets of transcriptional.
Okay, of course, this is highly advanced. Yes, now it is just showing you that it is far more complex that we are talking about. Yes, generally, nearly everything can happen at once or nothing. So it is highly complex, but this part you do not need to understand generally. Just get the idea that it's very complex, yes?
Machinery can move in the same direction to create many copies of RNA simultaneously. But when multiple transcriptional machines move in opposite directions, interference occurs. There are three main types of interference that can occur between pairs of transcriptional machines by machinery that is already transcribing. It is the combination of interactions in the system that allows the cell to regulate what RNA is produced. This allows the cell to respond to changes in its environment and to function. Without transcriptional regulation, life would cease to exist.
Okay, and it's enough. Thank you very much for your attention.